Hi, everyone. My name is Dr. Siddharth Prakash at the University of Texas Health Science Center at Houston. I am here going to tell you about a new research project that we're starting to understand why people with Turner syndrome are born with certain types of heart and blood vessel problems. My co-worker in this effort is Dr. Casey Gifford at Stanford University, and she contributed much of what I will be describing. A little about me. I'm a cardiologist in Houston, Texas. I've been long interested in genetic causes of bicuspid aortic valve and other kinds of heart and blood vessel problems that are related to bicuspid aortic valve. And I'm co-director of our adult Turner syndrome clinic, along with my colleague, Dr. Michelle Rivera. We see patients from all over Texas and the surrounding states. We set out to answer a few research questions that came about because we wanted to learn why certain types of heart and blood vessel problems are so common in Turner syndrome. For example, do some or all cells in the growing embryo need to have two X or Y chromosomes in order to form a normal heart? And which cells in the embryo actually need these chromosomes to function normally? What is the lowest percentage of cells with two X chromosomes that are needed to form a heart, or an X and a Y chromosome? We also know that not all people who have 45X karyotypes in their blood cells have heart problems. So what other factors besides having a 45X karyotype are needed to develop heart abnormalities in Turner syndrome? To answer these questions, we developed a method in our laboratory to create stem cells from Turner syndrome patients. This slide shows you how we do it. Using a regular type of blood draw from a person's arm, we can take peripheral blood cells and through a process called reprogramming, turn them into stem cells. These stem cells are pluripotent, meaning that they can turn into any cell type in the body. Through a process called differentiation, we can order them to become cells that we want to study, like heart valve cells or heart muscle cells. We can also use a different kind of method to create organoids, or miniature organs in a dish that we can study to learn how organs develop. In this case, we'll tell you about a process that we use to create heart organoids. If we combine cells in a certain way, we can form a beating heart in a dish. The heart cells have all the different types of cells that we see in a normal heart, but they form in a matter of days instead of weeks or months, as in a developing baby. If we take cells from people with Turner syndrome who have mosaic karyotypes, the cells in the organoid that we are studying also have mosaic karyotypes. This allows us to determine what mix of cells is necessary for a heart to develop and what type of cells in the developing heart function normally or abnormally in Turner syndrome. This project is just getting started. It would not have happened at all without the support of several people that I want to thank. In particular, Stephanie McDaniel and the Graham McDaniel Foundation, Cindy Skurlock, and the Turner Syndrome Society of the United States. With their support, we hope to have answers to our questions soon and be able to share these with the Turner syndrome community. More importantly, we think that these answers will help us to develop new treatments for heart problems in Turner syndrome in the future. Thank you, and please participate in these and other studies on Turner syndrome.
Hi, my name is Rebecca Nickmeyer, and I'm a professor at Michigan State University. I'm here today to tell you about our study, which is focused on identifying genes involved in the neurocognitive profile of Turner syndrome using exome sequencing. Now, while most people with Turner syndrome are of average or above average intelligence, Turner syndrome can affect specific areas of thinking and learning. For example, many women and girls report challenges in the area of attention and executive function. On an everyday basis, that could mean difficulty getting and staying focused, difficulty planning and making decisions, Challenges inhibiting intrusive thoughts and behaviors, trouble doing multiple things at once, or holding information in your mind's eye, like remembering a shopping list as you go through the supermarket. Some women and girls report issues with visual spatial and visual motor functions. What this looks like would be difficulty understanding things like maps and charts that rely on visual information, trouble understanding where your body is in space and in relation to others, as well as trouble doing activities that need hand-eye coordination, like riding, bike riding, or driving. Some women and girls with TS report problems with social cognition and communication. So this would manifest as difficulty understanding what other people are thinking and feeling, and that can translate into issues initiating or maintaining peer relationships. Finally, many women and girls report challenges with mathematical reasoning. Math does matter every day, so this could manifest itself as trouble managing money, telling time, recognizing and remembering numerical patterns like your PIN number for the ATM, challenges interpreting graphs, charts, and tables that rely on numerical concepts, as well as participating in certain sports, games, and hobbies that might have a strong math component. Now, there are general interventions that target these areas. For example, stimulant medications, originally developed for ADHD, can help with attention and executive function. There are also training programs to improve things like working memory, mathematical skills, and social cognition. But there are no TS-specific therapies for thinking and learning and challenges. And that's really because scientists don't understand why those challenges occur. Scientists think these issues happen because of a missing chromosome, but we don't know which genes on the missing chromosome are most important, and we don't know how those missing genes interact with genes on the remaining chromosomes. We also don't know why some women with TS might have problems in many different areas, while some might only have a few problems and others no problems at all with thinking and learning. Now, exome sequencing is a way to study DNA, focusing on the most important parts called exons. Exons tell our cells how to make proteins, which do most of the work in our bodies, including in our brains. So our goal is to identify genes involved in the neurocognitive profile of Turner syndrome by integrating exome sequencing with cognitive testing. So who can join this study? Right now, the study is open to women enrolled in the Turner Syndrome Research Registry who have also donated DNA samples to the GenTech Registry. But we are hoping to expand the study in the near future to involve other women in the registry who are willing to donate new DNA samples for genetic testing. What do study participants have to do? Well, first, they have to complete some cognitive tests and surveys on their computer. And second, they need to give the research team permission to access their genetic information. How do people join? Eligible women will receive an email inviting them to participate. So check your email. If you've been invited and you're interested, all you have to do is follow the link provided to consent, and then we'll send you the link for the cognitive testing. Other things you might want to know: this is a voluntary study, and all the information gathered is confidential. Participants are paid. They get $20 as a thank you for participating and to compensate them for their time doing the cognitive testing. If you're interested in learning more about the study, you can reach out to our study office at 517-355-3977. I want to end by talking about how we hope this study will benefit people with TS. So first, by identifying genes and genetic variants that impact thinking and learning in Turner syndrome, we hope it will help us predict problems before they emerge. So the typical neurocognitive profile usually comes out or starts being noticed when kids are school age. But a lot of brain development has already happened before that. And in fact, a lot of my work has been looking at early changes in the brain of infants with Turner syndrome. So we know infancy is a really critical time for intervention because the brain is growing so quickly. So we hope in the future we can use this genetic information to develop interventions focused on infancy and toddlerhood prior to the emergence of symptoms so that we can reduce or prevent them entirely in the future. Second, by identifying genes and genetic variants that impact thinking and learning in TS, could help us actually develop new medications targeting these areas that are really specific to TS, as opposed to the more generic treatments and interventions that are currently available. So I hope you've enjoyed learning about our study, and if you're eligible, I hope you'll consider participating. Thank you so much for your time.
All right. Hi, everyone. My name is Shanley Davis. I am a pediatric endocrinologist at the University of Colorado and Children's Hospital Colorado. And I am presenting today on our proposal for a rare disease clinical research consortium project that a lot of people are involved in. So I'm presenting on behalf of kind of our whole group here. Our proposed name is called NASCAR, Networking for Advancing Sex Chromosome Aneuploidy Research Readiness. And just to back up a little bit and just make sure everyone's on the same page, Turner syndrome is, when you think of a broader picture, part of the sex chromosome aneuploidies, so a different number of sex chromosomes other than the standard 2 sex chromosomes that you see typically. So you can see here, when we look at all of the sex chromosome aneuploidies, these top 4 here are including karyotypes that represent Turner syndrome. So a little bit of background in the Turner syndrome, as well as just the greater SCA community, we have a significant unmet need for evidence-based clinical treatments. We have many great clinicians and researchers across the country with a lot of great ideas. We also have strong partnerships with advocacy organizations, including TSSUS and other Turner syndrome and sex chromosome aneuploidy advocacy groups, as well as the patients and families more broadly. And we also have a basic infrastructure with a clinical research longitudinal registry to collect natural history data for these conditions. However, we have not a lot of money, and funding would help us really organize and advance the research potential and potential for treatments in these conditions on a broader scale. So there is this NIH funded resource called the Rare Disease Clinical Research Network, or RDCRN. And this funds approximately 20 different RDCRCs, or the consortia, for specific rare disease groups. This is just this honeycomb shows some of the different currently funded RDCRCs out there. Every 5 years, this funding source opens again for new rare disease research consortia, and we have submitted to be a part of this. Each of these rare disease research consortia are a collaboration of clinicians, researchers, and other individuals in the stakeholders invested in these rare disease conditions, with the goal of trying to get us to a place where these rare disease conditions can be what we call clinical trial ready. And we indeed want to be one of the new RDCRCs that will hopefully be funded this year. So backing up a little bit of clinical trial readiness and what does that mean for a rare disease, there's lots of different components that have to be put into place in order for us to be able to do clinical trials for treatments in rare disease conditions. We have to know the natural history of the rare disease and what we specifically want to target changing. We need to know the priorities of the community from a research perspective. We need to have valid outcome measures to measure what's important to the community in a way that we can actually test differences if we are doing an intervention of some sort. We often need biomarkers because often the outcome measures that we care about are down the road many, many years, and biomarkers may be used to be able to identify changes in these long-term outcome measures in a much more compact clinical time frame. We obviously need treatments and pilot studies as well to be able to get us to a place where we can conduct large-scale clinical trials. Our proposal has leadership proposed from different sites throughout the U.S., different specialties, and different investigators at different organizations and targeting different conditions overall. So the administrative core is the overall PI is Nicole Tartaglia, who is a developmental and behavioral pediatrician who has worked in sex chromosome aneuploidies, both Turner syndrome and other X and Y variations for her entire career. The Career Enhancement Core is being led by Judith Ross, who is a pediatric endocrinologist at Nemours Hospital for children in New Jersey. She has also been heavily involved in clinical trials for Turner syndrome as well as other X and Y variations. The Pilot Grants Core is being led by David Hong, who is a psychiatrist at Stanford, who has done a lot of both clinical but also primarily research for Turner syndrome and these other conditions, looking at differences in brain development in particular. And then the 3 projects that we're going to talk about a little bit more on the next page here. So project 1 is longitudinal registries to inform the natural history of all SCA conditions. So this is building up our existing registries that we have called Insights for Turner syndrome and Galaxy for other X and Y chromosome variations. These are clinical registries that are at 16 different clinic sites throughout the US, and individuals with conditions, we ideally want to get a representation of individuals with SCAs that are appropriately representing the entire population across the US. And this can serve as an infrastructure for natural history questions as well as future clinical trials. In Insights in particular, we currently have 700 participants with Turner syndrome at 13 different sites. This thus far has already led to 3 publications and has really been built on the support from community organizations. Project 2 is being led by Dr. Strang and Dr. Prakash. Dr. Strang is a neuropsychologist at Children's National who has an interest in and has a lot of existing research looking at individuals with X and Y variations as well as intersex conditions that really using community engaged participatory research methods. And Dr. Prakash, as you all know, is a cardiologist at the University of Texas at Houston and has had a longstanding history, particularly in Turner syndrome and engagement with the Turner syndrome community. The Project 2 goal overall is to engage the SCA community to identify and prioritize clinically meaningful outcomes for this population that we may target in future trials. So essentially understanding more what is important to the community for us to be studying. And then finally, Project 3 is looking at establishing severity scores and predictions of those severity scores for individuals with sex chromosome aneuploidy. The focus on Turner syndrome and Klinefelter syndrome is the 2 largest groups that we are going to start with studying. And this is being led by Dr. Armin Rosnahan at the NIH. He is a neurologist who has been at the NIH for many, many years and has a robust research program that has been studying, looking at RNA-seq and other multimodal technologies in order to advance research and biomarkers in these conditions. And so for this proposal, we aim to use Insights in Galaxy data, including the clinical records and the biorepository, in order to be able to define what we call dominant axes of clinical variation. So essentially developing these clinical severity scores, as we know that these conditions exist on a spectrum and not everyone is as complicated or as involved as others, and to develop then biomarkers that might help us predict these severity scores in this population as well. This is not an exclusive group by any means, and if individuals are interested in being involved and not already involved, go ahead and email me and we'll be sure to find a place for you to fit into this future if funded. Thank you.
I'm Melissa Aldrich at UT Health, and I have to make a quick disclosure that you'll see a movie or 2 using something called near-infrared fluorescence lymphatic imaging, and it uses off-label use of indocyanine and granadine. So let's see if I can get this to progress. Here we go. And so I'm astonished. I'm not a Turner syndrome researcher at base. I'm a lymphedema researcher, but I was astonished to find there are over 85,000 patients with Turner syndrome in the US. and about 3/4 of them will present with puffy hands and feet at birth, which is lymphedema. It seems to disappear, quote unquote. We don't really know that it does during childhood for some of them. And even though I just saw Michelle's wonderful slides and 33% report they have lymphedema along with Turner syndrome, a small study has shown that if you use this, see this grainy picture here, it's a type of imaging called lymphoscintigraphy, that actually closer to 80% of adults with Turner syndrome, despite what they believe, they actually have lymphedema. They cannot even move contrast dye up their legs as lymph would move. So there's not very much research done on lymphedema within the Turner syndrome community, and we'd like to change that. Just show this because a lot of people are very familiar with veins and arteries, but the lymphatics are your third highway system for removing all of your waste. Here I just show that you have some primary pre-collector, collector type, like tiny, medium sized and larger vessels in your body. They're everywhere. They're in, it includes your bone marrow and your thymus, where you make all your B cells and T cells. And it includes your spleen, it includes all the little villi in your digestive system and all the hundreds of lymph nodes in your body. So it's a huge system. And really, there's not much study of what it does or how it works in Turner syndrome. So I work with a group of engineers who use this technology that can kind of see through your skin and show you, at least for the lymphatics closer to your skin, whether or not they're pumping or they're moving and where they're located and how they're anatomically arranged. So we call it NERFLY for short, and it just uses near-infrared fluorescence lymphatic imaging. And it also goes by ICG lymphography using some other systems that aren't as good as the one that I get to use. So it has this kind of boxy looking thing you can see here in the middle that holds computer components and laser diodes and so forth. So what we do is we inject a little dye near the wrist, say for an arm. We can also look at legs and tummies and all sorts of things. We shine a laser diode that uses a wavelength that's near infrared, and I won't have time to show you, but if you get a flashlight after this and shine it through this part of your hand, you'll see a red light coming through, which is not your red blood making it look red. That is the red light that just doesn't get stopped by melanin or hemoglobin or water, things that make up tissue. So we take advantage of that, and the dye that we use happens to fluoresce. Once it gets in your lymph vessels, it fluoresces at that wavelength. We capture it with special night vision enabled technology, night vision technology enabled camera systems and collectors, and we can get movies out of it. So here's actually a physician being imaged with these cameras hanging over her arms. And here's what we see. We see, let's see if I can make it go here. We see lymph going up from her hand, which would be up at the top there, through the collectors or the large vessels into her lymph node basin in her armpit. So that's what you want to see if you have healthy lymphatics. So if you don't have healthy lymphatics, you see all kinds of things like this, where we'll inject dye and put some black tape over it because it's so bright for the cameras, and it doesn't move. It doesn't move up those vessels like you just saw. You'll see some punctate things here that I think look to me like tertiary lymph nodes, like you might see in rheumatoid arthritis. You'll see twisty, tortuous vessels as you see here and here. So we can tell if you have lymphedema based on this imaging thing. So we're hoping we get millions of dollars to look at Turner syndrome, lymphedema, and kind of on the side, learn a little bit more about other things of Turner syndrome and build a little bit of build up a biobank a little bit. So we're proposing taking blood from 12 karyotype Turner syndrome subjects that'll be taken to be pluripotent stem cells and then differentiated into what we call lymphatic endothelial cells. And then we want to see what genes are upregulated, what genes are downregulated. We want to see if there's something around the DNA, the epigenetics is what they call it, that's keeping things from being expressed. And see if these blood cells from those patients can actually develop in a Petri dish into the little lymphatic capillaries like normals can. And if not, what's different? That would tell us what proteins might be missing or might be able to be treated with a drug later, label drug. The other thing we want to do is we want to use that imaging I just showed to see what the layout of the vessels looks like in Turner syndrome, because no one knows. And at the same time, we're going to take blood, and so it's going to look kind of complicated, but we're doing a lot of things for a lot of money. We want to see what their T cells and B cells are looking at, because, Turner syndrome patients have a 2 to 3-fold higher likelihood of developing something autoimmune. And we suspect that kind of more a garden variety of lymphedema is autoimmune. So we want to see, is that what's going on with Turner syndrome? We don't know. We're going to enable this with artificial intelligence and highfalutin bioinformatics using an expert down at UTMB to get all of our large data into a digestible format so that we can find out if there are targets for helping with lymphedema in Turner syndrome, with patients with Turner syndrome. We even have a group in the Northeast who can do an inverse vaccine. So if we find something is autoimmune, they have a way to very simply and almost naturally reverse that autoimmunity, and it could be a cure. So those are our pie in the sky hopes with this research. We want to image at least 10 patients with Turner syndrome, because we don't even know if the layout of the vessels is similar to what we see in other humans. So normally you'd see vessels coming up along the back of this thigh and maybe going through the ventromedial bundle or whatever. You can see here, this is not a patient with Turner syndrome image over here, but this looks like, I don't know, a maze almost. Lymph is moving up this patient's legs. She has a type of primary or inherited lymphedema. And until we saw this video, we had no idea that vessels could look that crazy looking, kind of. And yet therapists who were trying to give manual lymphatic drainage were brushing with the massage strokes this way. And you can see that that wouldn't be a personalized treatment, nor would it be effective. So we will get that information as part of our project, along with the genetic and the proteomics data. And we're hoping and keeping our fingers crossed that we can obtain this funding. And I just put this slide up to show that I'm just part of this group of my boss Eva Sebek and John Rasmussen developed the imaging that we want to use. Other people here are all part of the team using it in other applications, but we'd really love to see something happen in Turner syndrome. Thank you.
My name is Michelle Rivera. I'm a pediatric endocrinologist here at the University of Texas Health Sciences Center in Houston. And today we're going to review the data from the third set of national surveys distributed through the Turner Syndrome Research Registry. The third set of national surveys, we started distributing these surveys at the national conference last summer. And it was composed of several different parts. So the first survey that we distributed was the introductory survey, and that was intended to gather demographic information such as age, economic status, karyotype. And based on the responses to that introductory survey, the participant was offered the opportunity to participate in several other surveys. So one was the general health survey. That survey was intended to collect current physical and or mental health data regarding their individual concerns. 269 individuals went ahead and answered that particular survey. The other option was also to participate in the perinatal and early life survey. That survey was intended to gather information from parents regarding the pregnancy of the child with Turner syndrome, as well. So it was going to gather information regarding their experiences, what resources were offered to them during the pregnancy, and then what was the infant and early childhood experience of that particular individual with Turner syndrome. So what types of medical conditions and other challenges that particular individual face. 265 individuals answered that survey. 82% of the respondents were white or European, 6.7% identified as Latino, Hispanic, or Spanish, 4.5% multiracial, 3% Asian. Other ethnic groups were represented at below 1%, which again highlights an area where we need to perform more outreach. This, unfortunately, is also what we see in our clinical practices, where the majority of the individuals that we see do not represent minority groups. Very few minority groups are represented in our clinics as well. So this is an area where we feel we need to improve our outreach as well. Of those who answered this survey, 71% were adults with Turner syndrome, and about 20% were caregivers or parents answering questions for their child with Turner syndrome. When we look at the responses related to certain areas of interest, we looked at the estrogen use in this population. About 80% of the participants indicated that they had taken estrogen at some point in their lives. About half were on estrogen for more than 10 years. Interestingly, we did see that the median age of initiation of estrogen therapy was around 14, which is older than what our current guidelines would recommend, but this might represent an older population answering these questions. So I think we'll hopefully see this number decrease over time. Of note, though, the median age of ceasing or stopping estrogen therapy was 40, which is much younger than we would recommend, which with the typical age of menopause being around 52 years, that's actually the recommendation. We are interested in exploring further what barriers exist towards the continuation or use of hormone replacement therapy. And we did have at the national meeting an option for individuals to participate in an estrogen experience survey, and we will be bringing those results to you soon. Other areas that we found that were notable in the responses. So in addition to congenital heart disease, such as bicuspid aortic valve and coarctation of the aorta, cardiovascular disease risk factors were pretty common in the respondents, with a large percentage indicating high blood pressure, high cholesterol, and diabetes, as well as a large percentage of individuals also indicated that they suffered from other concerns such as lymphedema, for which we need to improve our screening and therapeutic modalities. Skeletal concerns were very common, with about 35% of individuals indicating they had low bone density or increased risk factors for osteoporosis or osteopenia, and about 11% indicated they'd actually suffered a fracture in their lifetime. Mental health concerns were very prevalent as well in the population, with the most common mental health concern reported being anxiety. And so those were the individuals that were diagnosed with mental health concerns, but over half of the people that responded to this survey also said that they felt they may have had an undiagnosed mental health concern. And so this tells us that we really need to do a better job of screening for mental health concerns at routine visits for our population with Turner syndrome. So jumping into the early life survey, we noted that in addition to the expected findings of lymphedema, cystic hygromas, cardiac and renal anomalies, a lot of our infants and children faced other challenges such as poor or slow growth. A lot of the children had ear, nose, and throat disorders that required surgical interventions, and also indicated developmental or behavioral concerns. So 70% of children required at least one surgery, and these respondents we were asking questions about the first 4 years of life. So 70% of these infants and toddlers required at least one surgical intervention. Many, about 40%, required a breathing tube at birth. Another 30% required a feeding tube. And then the parents were often felt unprepared for these challenges. About over half of the study participants indicated that they received no resources about Turner syndrome prior to the delivery of the baby. And 80% reported that they felt their prenatal providers had little or no knowledge about Turner syndrome. Only about 15% were referred for genetic counseling. Most of these individuals were seen at private OB clinics or community hospitals. So a few take-home points. There's a large need for improved education and communication with our prenatal providers. Children often faced lots of challenges and required services such as individual learning plans, 504 plans, physical, occupational, and speech therapy. And parents require ongoing support and access to resources to allow them to help their children with their feeding, hearing, vision, growth, and developmental concerns.
Hi, I'm Tamar Green from Stanford, and I'm associate professor. I'm a child psychiatrist by training, and I'm very kind of like excited to be here. I did most of my postdoc training in Turner syndrome. Then in my independent studies, I started to study other conditions. And what I'm going to talk about with you today is I'm going to introduce you our new idea for research, but I'm not going to introduce you any results of that because we haven't started it yet. But I'm kind of going to go over the rationale in why we want to do this work. And this work is also inspired by my many interactions with older women with Turner syndrome. I usually study kids, and I used to get like constant requests that will also study adults, and specifically symptoms of depression and anxiety. So it's kind of like my kind of like coming back to it with like hearing what the women with Turner syndrome really like to us to study. I'm going to start with kind of like the concept of why I do what I do. So this kind of like phrase talks about how we need to exercise justice based on the sexes, and I'm just gonna I kind of like tweak that to say that medicine should also be exercised this way, and thinking about differences in sexes and how we treat different people with different needs. And what kind of like when I started to think about this was in a review I conducted in 2018, thinking about sex differences in psychiatric morbidity. And as you can see on the slides, on the left, we have this discrepancy between the sexes when we think about neurodevelopmental conditions like intellectual disabilities, ADHD, and autism. We see higher rates of males versus females that are affected by these conditions. But by adulthood, these conditions also affect kids, and by adulthood we see something different. We see more and more mood and anxiety disorder, with females being much more affected than males. And specifically in conditions like Turner syndrome, and specifically to Turner syndrome, there's effect on that we aware of on verbal and IQ. We know that there's some strength for the girls with their verbal IQ, and some weaknesses that are so that will be difficulties with emotion processing, lower executive function skills, and lower visual spatial skills. And here I'm gonna just briefly kind of like talk about a study that I did when I was a postdoc that took and compare whether girls with Turner syndrome have similar or different ADHD symptoms of attention to children with idiopathic ADHD. And what we found is that 50% of the girls with Turner syndrome will score over the at-risk for attention difficulties or for and 25% of them will score in the clinical significant range. And girls with Turner syndrome and ADHD were as hyperactive and inattentive as boys, while the girls with Turner syndrome and ADHD were more hyperactive than girls with this condition. So overall, we see that there's much higher risk for ADHD in this population. And we do know from the literature that neurodevelopmental issues without Turner syndrome put individuals at risk for depression and anxiety. And depression in individuals with Turner syndrome is actually we start to see it in a pretty young age, and it's just going and becoming more and more prevalent. And we do need to think about the fact that Turner syndrome is associated with one X chromosome, but also changes to sex hormones. So that also might be another mechanism that kind of like related to depression. There's other people who think that maybe the short stature is another risk factor for that. And just kind of like to illustrate what I was talking about before, is that what you can see here on the x-axis, we see three groups, girls that are younger than 11, individual teens between 11 and 18 and over the age of 18. And Morris et al. Showed that depression levels increase in this across this age group and reach to that 75% of the individuals that were asked had higher depression level compared to controls. It doesn't mean that 75% of them had depression, just that it's for 3/4 of the individual, they had higher symptoms of depression when tested. When I conducted this review, what I also kind of became more and more aware of is that individuals with Turner syndrome are at much higher risk for variety of autoimmune conditions. Of course, I think many are aware about for the thyroid issues, but also celiac disease is very common, problems with your skin, and also inflammation of the GI system. So this combination of understanding that there's higher rates of depression and also higher rates of autoimmune condition led me to kind of like the hypothesis that the immune system might have play a very significant role in these higher levels of depression, and might also gives us an avenue of how we want to address depression in Turner syndrome, circling back again to being like very specific and thinking about each individual and why things are happening. This is might be very obvious in other conditions in medicine, but not in psychiatry. In psychiatry, we do still have like this one size fits all. So kind of like thinking about it in a more specific way. So what's next? We just got our IRB approval to collect preliminary data. And we are now recruiting 10 women with Turner syndrome. I put our QR code for our lab website, and it's not up yet because we literally got the approval on Friday. So so more to come, and I'll be happy kind of like to share once we active, and yeah, I'm looking forward for that.
My name is Michael Silverbach, and I want to tell you about personalized valves that we have designed that may be valuable to the Turner syndrome community. Just to briefly begin, I think I don't need to tell this audience the significance of the bicuspid aortic valve in Turner syndrome, but I'd like to take a moment to point out a few things that maybe nobody has really thought much about. Here we have the normal configuration looking from below of the aortic valve with 3 symmetrical and equal size leaflets. And down here we have a typical bicuspid aortic valve, which we often call the left-right fusion type. But the thing I want to point out to you is that, and which people don't pay much attention to, is the asymmetry of the aortic sinuses, this housing that surrounds the valve. And this is quite typical of bicuspid aortic valve, with 2 small sinuses and 1 large sinus, even though there are only 2 functional leaflets. And the problem is that, these valves often thicken and need to be replaced. And we got the idea that it may be the shape of the aortic sinuses may be critical in the nature of the flow that comes out of these valves, particularly when they're replaced by a surgeon or an interventional catheterizer. So if you could imagine, if typically when the surgeon or the cardiologist goes in, they go over the shelf and they put in a symmetrical valve. But what would happen, and this probably happens to everybody who has this type of configuration, they'll put a symmetrical valve into a situation where there are asymmetrical sinuses. And we hypothesized that the result of that would be very abnormal flow downstream of the valve, and may also lead to the early senescence of the valve itself. So we invented a new kind of valve that I want to tell you about. So we are very interested in how the flow might be affected if we put matching valves that match the shape of the aortic sinuses. And so I'm asking the question here, what does an Urban Outfitters sale have to do with aortic wall stress? And I'm going to show you this little video from YouTube. And what you're seeing is a bunch of people that are standing outside. This is the door here, and this door is going to rise up, and there's a bunch of people, and they're murmuring, and they're very quiet. And then as we move along here, as the door begins to open, there's a cacophony of people, and the energy is just building, and they're bumping into each other. This is sheer stress within the crowd of people. Now, as this door opens, the noise will become deafening, and they start to run through this door, and you're going to see that as well. And I want you to imagine something, because it'll be important for a video that I'm about to show you. If we put little lights on all these people's heads, and then we turned off all the lights in the room, we would see streamlines of all those little heads as they move through this door, as they rush with great excitement and energy through the door. So we're going to watch that. Here they go. They're screaming. They're screaming. Here comes the doors going up, and look at them run. Okay, and right here, I want to point out something. There's a post here. And I'm going to keep it moving along. They start spinning around the post and hitting with the shoulders. So this is shear stress within the vessel of the aorta. But what's happening along this post and at the edges of this opening valve, if you will, a lot of turbulence and disrupted flow of, in this case, Urban Outfitter exuberant buyers at this sale. But we're going to think about that as what happens when flow is made abnormal as it moves through a valve. So there they go. See them bumping into that post and how they're spinning. And then they start speeding up. but they're going very slowly through here as the energy gets absorbed by the wall and these obstructed things in their way. So I know that's kind of a unusual analogy here, but I think it's quite appropriate for what we're talking about. And so here is an aorta from a 43-year-old woman who has bicuspid valve aortopathy with this tremendously dilated aorta. This woman doesn't have Turner syndrome, but this is very typical of the aneurysmal aorta. And here is our valve placed in the sinuses of this person who has this very typical asymmetrical aortic sinuses. And here is the experiment that we did. We did a flexible 3D print of this individual's aorta. And then we made a valve using our patented technique. And we've placed a valve that reflects the shape of the aortic sinuses, one large sinus and two small sinuses, just like I showed you in that first picture. And this is one experimental design. This is our test. And this is our control design. We took that exact same aorta, but instead of putting a matched sinus, matched valve with the matched large sinus and two smaller valves with the two small sinuses, we put a symmetrical valve into this exact same aorta. Note that there's a large sinus and two small ones, just like I showed you. And then this is a bioreactor that's all made out of plastic because we're going to put it into an MRI machine that has 30,000 force of gravity. So there can't be any metal on there. And there's some tubes here that go out to our control room so we can pulse and make the simulation of the heart circulation. And the two valves, the same aorta with the control and the test valve are inside there. Now here's the actual, box cutter, we call it, this device that the surgeon or interventional catheterizer will use. This is the one that will make the large valve leaflet, and this will make the two smaller leaflets, and then it could put in an armature and then placed into either a delivery device in the cath lab, or the surgeon can then put it into the aorta. So that's our project. We're pretty excited about it, and really want to thank everybody that's helped us with this project. The Turner Syndrome Society of the United States, of course, the Revell Pittman Fund for the Turner Syndrome Society, our own institution, the Friends of Dornbecker, and the Dornbecker Children's Award that I received to work on this